receptor interactions and how it results in hormone action. To understand her lecture, I will give a brief introduction to receptors in this lecture. And therefore, in that process, I will be summarizing animal endocrinology in about 10-15 minutes. So the three things I want to achieve: extend Professor Yogendra Singh's lecture, make you understand Rita Singh's lecture next after me, and also summarize whole of endocrinology with about 10 slides. I hope I will achieve this. Now, the first thing is, of course, I don't like talking to computers, so I want to make it a lighter vein. This is essentially a picture which tells you that this is fun, but I still prefer seeing you all at the old watering hole. Even the crocodile does not want to talk to the computer.
Sir, share the screen, sir.
I have already done that.
It lost and again you have to do it.
My God. Thanks. Can you see it?
Yes, yes.
Okay. So we come back to this lecture. I hope I am audible. I hope you can see the slides.
Yes, yes.
Okay. The first thing is in the lighter vein. I don't like to talk to computer, this whole online business, but then we have no choice. And therefore, in a lighter vein, I am showing you this cartoon where even the animals don't like to speak to computer. They want to meet at the old watering hole. Anyway, we come back to our thing. Yesterday, Professor Yogendra Singh talked elaborately on protein structure, especially on post-translational modifications. I'm just summarizing this. Proteins are polymeric macromolecules made of monomeric amino acids. Simple proteins are made of only amino acids. Conjugated proteins have, in addition to the apoprotein, other biomolecules like sugars for oligosaccharide in the form, lipid, metal, chromophores, porphyrins attached to the apoprotein. During biosynthesis, the growing nascent chain protein undergoes co- and post-translational modifications. Co-translational modifications are sensitive to inhibition by cycloheximide, whereas post-translational modifications are not sensitive to cycloheximide. N-glycosylation of asparagine is a co-translational modification, while O-glycosylation of serine, sulfation of tyrosine, which we discovered long back, acetylation of histidine are post-translational modifications sensitive to cycloheximide inhibition. There are over 240 reported post-translational modifications. The students should remember the great area and the enormity of the information available. I will focus only on one of them, so-called phosphorylation and dephosphorylation at the serine residue, and see what is its importance in the regulation of metabolism, especially regulation of blood glucose levels. For that, I will have to explain the context, and that means I have to summarize endocrinology very briefly. We are talking about hormonal regulation of blood glucose, and therefore I should tell what these hormones are. Hormones are communication messengers in animals as well as in plant systems. Even in microbes, there are other types. And these hormones are essentially were discovered long back in the case of animal endocrinology. Official historians give the credit to a person called Starling, who discovered the gastrointestinal hormone in 1903. Why we are recollecting this? It is extremely important because this is the only method available to discover hormones. This is called technically surgical extirpation followed by replacement therapy. What does this mean? You remove some part of the tissue in the body which you suspect to be making the hormone and see what happens to the rest of the body. Record all the physiological damage that it does. And now either put back the tissue that you have taken out, or make an extract and then inject it to animal. And now you see that all the deficiency symptoms that you created are restored back to normal. That means that tissue which you removed is an endocrine gland and it secretes something called a hormone. He used the term hormone, which literally means to excite. Hormones essentially excite the rest of the target tissues, and that's how endocrinology started. Till today, this is the only method to discover hormones. However, if you read the subject well, you notice that almost 60 years before Starling, in 1839, another fellow called Berthold did a very simple experiment in chicken. He took young chicken, male chicken, and allowed them to grow into adult male birds. And most of you are familiar that these adult male birds have two features which all of you have seen. One, on the head, there is something called crown, and in the neck region, there is something called wattle, loose skin. In the second experiment, he took another group of young male chicken and removed the testis. This is called archiectomy, technically, and allowed them to grow. Surprisingly, they grow into an adult bird, but you don't see them sexually mature. They neither exhibited crown nor wattle. Then he did a third experiment, which is very simple and very elegant. He took the testis that he removed in the archiectomy process and put it back in a peritoneal wall, totally different from the original place of testis, and then allowed the birds to grow. Surprisingly, both the crown and the wattle came back. And that is why he said there is a communication from testis to the head region and to the neck region. He didn't know how this communication went. It could not have been nerves because he had removed the nerves. And therefore, it is believed that he discovered the hormone, but historians don't give any credit to Berthold. It doesn't matter. So from the days of 1903, Starling, who discovered this gastrointestinal hormone, many people repeated this experiment in hundreds of animal species, discovered all the hormones that we talk today in any textbook of animal endocrinology. Now we jump. What happens? What do these hormones do? When you look at this, we notice that hormones have structures. One, that means chemically they represent diverse structures. We will not go into detail. Some are amino acids, some are proteins, some are polypeptides, some are steroids, some are fatty acids. All sorts of chemistry is represented by diversity of hormones in the so-called animal kingdom. The second thing is that they act. I am temporarily using the word hormone action. Later we will realize hormones don't do anything. They don't act. This 100 years it took to realize this truth. So what do these hormones do? Every hormone action involves some stimulation of a physiological event. Let us say growth hormone stimulates growth. Let us say thyroxine stimulates basal metabolism. Let us say estrogen stimulates growth of accessory reproductive organs in the female mammal. and so on and so forth. Every hormone has an action, but the most surprising thing is that this action is extremely specific. On some other occasion, I mentioned that you look at any physiological process, any developmental process, any behavioral process, you notice one feature, an extraordinary specificity. Biological responses are highly specific to particular stimulant. In this case, the stimulant is a hormone, and the response is in a particular tissue called target tissue. So this gave rise to the second feature of hormones, that in their action, there is an extraordinary specificity. Moreover, there is another feature of these hormones. If you measure the concentration of circulating hormones, these are in nanomolar range. That means 10 to the power of -9 or even less. However, if you look at hormone effect, it is very, very loud and dramatic. In other words, hormones hardly whisper, but you see dramatic effects. If you see a young calf grow into a magnificent buffalo or a horse or elephant, you can see that this whole miracle of growth, perfect growth, was brought about by only few micrograms of growth hormone. In other words, extremely low circulating concentrations of hormones bring about changes which are very dramatic. Hundreds of examples we can quote on that. Similarly, if you take in the case of amphibians, you understand the life cycle, and there is a stage called tadpole. Now, this tadpole becomes adult frog. Frogs and tadpoles don't look similar. There are dramatic changes in many parts of the body, and the tadpole becomes frog. However, you will be surprised that this, which is called amphibian metamorphosis, is brought out by hardly a couple of micrograms of what we call thyroxine, or a thyroid hormone. And therefore, people realized two features of hormone action. One, that they occur in extremely low concentrations, but they act on distal tissues and bring about dramatic effects. And therefore, we compare hormones to actually like a postcard. You originate, the postman travels very slowly, brings it to the house, and the effect is also very dramatic when you receive the postcard or Indian letter or any such thing. However, exactly opposite of this are the nervous system, neural system, where you have neurotransmitters which are secreted in high concentrations at the synapse, and they travel along the axon and produce their effect, very dramatic effect also. But then the effect is very alarming. So we compare neurotransmitter action as telegram, whereas hormone action we compare as postman system or postal system. And therefore, nature of hormone action, I am summarizing whole of endocrinology. Basically, they represent diverse chemical structures. So the name of hormone is not based on chemistry, but based on what do they do or activity. And their hormone action is characterized by specificity and occurrence in extremely low concentrations. And the third thing that we understand is it leads to regulation of metabolism. And people for the last 100 years have called hormone as a signal. Most of you are familiar with this signal engineering departments. They use this signal, means it is a coded information. It is, unless it is received and decoded, the signal will not be understood by everybody. For example, if army is moving in an enemy territory, the army people, the foot soldiers, will give signal. And who will understand this signal? The headquarters. But this, the enemy will not understand, because the language is a code. Therefore, in the same sense, hormones were called as signals because what they do, nobody had understood. And therefore, they said hormone is a signal. So last 120 years, we are asking, is this really true? What actually is the signal in hormone action or in endocrinology? We don't have final answers for this. Therefore, endocrinology is total information that we have to answer three questions. What is there a structural basis for hormone action? First question. Second question, how do the hormones bring about those dramatic effects in low concentrations with extraordinary specificity? And the third question is, what actually is the signal in endocrinology? The answers to these three questions, which are listed here, essentially form the bulk of endocrinology textbook. 130 years of endocrine research I have summarized in this slide. I will take 5 more minutes to understand each one of these answers. If you want to answer the first question, remember biologists ask this question always, does structure subserve function? It can be leaf, it can be liver, it can be animal shape, it can be anything. Biologists are obsessed to relate structure to function. Same thing happens in endocrinology. And to get that answer to the first question, I called it concern one. What do endocrinologists do? They purify a particular hormone. They make sure that it is pure. I use this word pure not in a religious sense, not holy. It means technically it is a homogeneous substance. There is no other substance other than the hormone. Then they determine the structure. This is, you need chemistry for this. And then they ask structure function relationship. They try to understand by chemical modification and then look at the consequence of that on hormonal activity. And then they get what is called structural clue to hormonal activity. Till today, we don't have structural clue to any hormonal activity. Hormones for therapy, because I said that yesterday that reductionist biology has one more theme in addition to understand nature, living organisms. It also wants to help mankind. Therefore, hormones are used as a therapeutic agents, and that is one of the applications of endocrinology. And for therapeutic use, you need to produce the hormone in large quantities, and that means technology is involved. Biotechnology comes in picture in the large scale production. If time permits, I will give an example. Especially nowadays, they use recombinant DNA technology, cloning and expression of these hormones. All right. This is all the work that last 130 years endocrinologists have been doing. For purification, of course, there are a variety of methods. Professor Yogendra Singh also referred to this. Biochemical methods, affinity methods, scale-up methods where chemical technology comes in. I also showed one example yesterday where a single-step purification of luteinizing hormone by affinity chromatography. I am showing the same slide. Therefore, let us say hormones can be purified, structure determined, and then you relate the structure and then try to get the structural clue to a particular specific hormone action. I also mentioned that the names of hormones are not derived by their chemistry, but by what they do or activity they have. And you measure the hormone quantity by measuring the activity that a given hormone exhibits. And therefore, this technique is called bioassay. Assay means estimation. You do bioassay means you are estimating biological activity using intact animals in the case of animal hormones. And here is an example of what bioassays we have, for a group of pituitary hormones called gonadotropins. Dr. Rita Singh will give more details in the next lecture. You don't have to, you just have to remember that bioassays are there. I also mentioned that for therapeutic purposes, you will have to produce these hormones in large scale by using recombinant DNA technology. I am not going into detail. I will give you one example. There is a hormone called prolactin. We have purified this prolactin from buffalo pituitaries through biochemical methods. We have also cloned the gene for cDNA for this hormone. expressed in E. coli because this is a simple protein. But most importantly, the recombinant proteins which are expressed in prokaryotic system or eukaryotic system do not fold automatically. You will have to do that in vitro. Protein folding is a very, very, very big technical problem. We learned the tricks, and here is an example of a recombinant buffalo prolactin folded, purified from the culture medium and folded properly. The biological activity of this in form of a dose response curve compared to the same biological activity exhibited by a native buffalo prolactin. That means buffalo prolactin purified from pituitaries of live animals or dead animals by slaughtering and through biochemical methods. Therefore, if you compare the two bioassay results, you notice that the dose response curves are almost similar. In fact, the recombinant hormone is more active than the native hormone. It means that we are able to clone and express these. Many people have done, and this is for therapeutic purposes. That is the summary of endocrinology. Now, when you look at that second question, and we said that hormone action occurs in low quantities, but the action is very dramatic, and there is an extraordinary specificity in its action. Therefore, people asked, what is the basis of specificity? Why a given hormone coming from, let us say, pituitary acts only on the gonadal tissue and not anywhere else? Similarly, for other hormones. And therefore, people were struggling to understand what is the basis of this specificity. For long years, endocrinologists were very silent because we did not know how to go about investigating. We learned these tricks from pharmacology, where people work on drugs. And drug action also is very specific, as all of you know, that you take for headache aspirin. Aspirin will not cure a skin disease. It will only cure headache. Therefore, in every drug action, there is a specificity. Therefore, people in pharmacology asked the same question, what is the basis of specificity? Then they discovered that the tissue on which a given drug specifically acts and exhibits its activity has a capacity to bind, recognize this drug, hold it for some more time than the other tissues, and respond to this drug. And because this is a property of only the target tissue, they said there is something in the tissue which receives the signal. They did not know the chemistry of that. They called it a receptor. So when this term was coined in 1930s, they only used it to represent an activity by which that recognizes the drug and responds to that in a specific manner. That's all. The activity is called receptor. But in order to know the biochemistry of this, people did very simple experiment. This also we learned from pharmacologists. This is called establishing the pharmacokinetics of a given hormone or a drug. What do we do, or what do scientists do? They take that particular hormone, pure hormone, attach a label, because hormones act in very low quantities. You can't see them, you can't measure them at that low concentration. Therefore, they put a label on that. What is a label? Label will amplify that presence of that hormone. It is like looking at a long shadow without looking at the person or anything, because shadow is larger than the person or the target, and depending on the angle of light falling on that. So therefore, when you look at the shadow, you believe that you are seeing the object. Similarly, in another case, for example, those of you who are aware of how tigers are counted, you hear all sorts of funny figures. Shah Rukh Khan will tell something, the Tiger Association of India will tell something else. How do they measure how many tigers are there in a given forest? All they do is they look at pub marks of tiger and believe that when they see the pub mark, there must have been a tiger. In other words, you measure the number of tigers by indirectly concluding it by looking at something else. It could be shadow, it could be pub mark. Similarly, hormones cannot be seen or measured, and therefore they put a label on that, which amplifies the presence. There are three types of labels: radioactive isotope, fluorescent compound, or an enzyme. By this, the hormone can be monitored. Now, if I take a radio-labeled hormone, inject it into an animal, it will enter the bloodstream, go through the bloodstream to all the tissues of that animal. And then if you plot how much of this label is there in each tissue versus time, including blood, you will get funny curves on this. All of them are bell-shaped, that it will enter the particular tissue and come out after some time, and that is called the dwell time. But among all the tissues, including blood, one tissue will exhibit a very strange curve, where when the hormone enters, that means when the label enters, there is an increasing in concentration, but it is retained in the tissue for a long time beyond the dwell time explicable. And then it slowly comes out of that. That means that particular one tissue among the 100 tissues has a capacity to retain, bind that particular hormone and hold it tightly, because there is a great affinity, and that tight binding will result in some action, and then it releases that bound hormone. And therefore, biochemistry people, pharmacologists, said that substance in the tissue, target tissue, which has the capacity to bind the particular hormone only with tight affinity, respond to that and then release the bound hormone, that substance is receptor. So the biochemistry people, when they use the word receptor, they refer to a chemical substance. When the pharmacologists use the term receptor, they refer to an activity of certain property. However, now slowly it is all clarified, and this experiment leads to the discovery of receptor. So in the same experiment, we can get the idea that this particular tissue has a receptor. Subsequently, if you take the tissue, grind it, and then do a subcellular fractionation, then you will also get an idea where that label is. Is it in the membrane? Is it in the nucleus? Is it in the cytoplasm? So you get a subcellular geographical location of the receptor. Subsequently, one can purify this receptor again by activity, and that becomes a pure receptor, and all receptors are proteins. And therefore, we have come a long way where we used this term pharmacologically for an activity. We use this term. The biochemists discovered this and showed it is a chemical substance, and hormone receptor interactions leads to subsequent activity. Professor Rita Singh will give you great detail about how gonadotropins, especially follicle stimulating hormone, interacts with its cognate receptors in the gonadal tissue. Those details she will tell. For the present lecture, this so-called hormone, we call it as signal temporarily, and there are a number of types of signals which living organisms respond. We all know light is a signal. Animals respond to photoperiod. The whole subject of chronobiology will tell you this. Green plants respond to light, and photosynthesis is a consequence. Of course, they are sensitive to different wavelengths of light. I'm not going into detail. What is important to know that we can sense light. Living organisms can sense light, and therefore light is a signal. Similarly, we can sense sound, hearing, we can all hear, and therefore this is one sensory perception, hearing. We can also pressure, we can sense. These are called mechanical receptors. In fact, the so-called sound is essentially listening to pressure in the air changes, and that when translated becomes sound. living organisms are sensitive to external voltage, electrical voltage. I will not go into detail. You know, there are the whole nerve conduction is that a responding cell is responding to external voltage, or there are ion channels which will respond to this, and therefore we can sense electrical signals. We can also sense chemicals in the environment, hormones, neurotransmitters, growth factors are some examples of chemicals. We also respond to pollutants, which are also chemicals. We respond to toxic substances in the food, water, and air. That is also a chemical. Therefore, living organisms can respond to these so-called 5 types of signals. And therefore, people have long told you that living organisms, especially mammals, have this kind of 5 types of sensory perception. But I must tell you now that we have another 7 types of sensory perception. We can sense the magnetic field of Earth and find a direction to a given place. That is called magnetoception. Birds, you must be reading in books, migratory birds travel 10,000 kilometers from Siberia and come to Agra. Similarly, many Chinese birds come to Kerala or some other place, and this bird migration, if you ask a simple question, how does the bird know where India is? It is all through to magnetoception. This is called direction finding. We also have some such thing. We also can, if you stand in a room or any animal, including a rat, it immediately knows what is the space available in that room. How did we measure all these things? We still don't have much detail about it. There are what we call space cells, place cells within our brain, which will give you an idea how far is this front wall, how big is this room or the space in which I am measuring. Similarly, we also know the time lapse. In fact, the biggest question in biology is how do organisms measure time and space? I'm not referring to wristwatches or clocks or calendars. I'm referring to how does our brain measure time lapse. There are time cells in our brain and they give us an idea about time lapse. Without going into detail, this is the biggest biological question. In developmental biology, you notice that a given species will develop only up to a particular size. You never saw in a developing rat or a human being a heart developing in the middle of a liver. Never. Which means the developing tissue or cells can measure space, limits of space available, as well as time. Gene expression changes temporally, and that means somebody is measuring the time. And therefore, we say the biggest mystery in biology, how organisms can sense time and space. Like that, we can go on. We can sense acceleration. So there are 7 more sensory perceptions in addition to this 5 that I have mentioned. Most of endocrinology, of course, is retaining only this, chronobiology or plant physiology, but all others people are not studying in detail right now. So that is an open area in biology. Once these signals are received by some receptor, these receptors will be now saying that they are molecules and they sense the presence of signal or stimuli and respond to that. So people then did a lot of experiments and asked, where are these receptors? What is the subcellular location within the target tissue? And they got answer. I'm not going into detail that there are receptors to water-soluble compounds or hormones on the membrane, because water-soluble hormones cannot cross the cell membrane. Most of you know structure of cell membrane. It is almost like a kerosene. It's a lipid bilayer where you see lipids as well as proteins. Now these membranes, which following the Singer-Nicholson model of fluid mosaic model, these has also receptor proteins which recognize water-soluble hormones coming from outside. For some other hormones which are water-insoluble, like thyroxine or steroidal hormones, the receptors are in the nucleus. Now, this itself has a big history. All of you must be reading newspapers. The present Vice President of USA, Kamala Harris, I'm sure you know that, her mother was an endocrinologist and her name was Shamala. She married an American, Garsky, and therefore her name was Shamala Garsky. She did PhD work with Garsky. She was my contemporary. She visited our lab also. And she reported, and with her boss, that the receptors to estrogen are in the cytoplasm of the target tissue. Another person, Elwood Jensen, also said the same thing, but Elwood Jensen got a Nobel Prize. Within 20 years, we know this whole work is wrong. Receptors are found in the nucleus, not in cytoplasm. However, it is very interesting that almost for the first time, the person who said that receptors to steroid hormones are there inside the cell was Shamala Garsky, mother of the current US Vice President, Kamala Harris. Kamala married West Indies person and Jamaican person, and that's how her name is there. But we are proud that Indians were involved in the discovery of these receptors. Fine. These two types of receptors are mostly proteins, and they bind this hormone or signal and then do something. This is an example of a receptor to thyroid stimulating hormone. Professor Rita Singh will give more details. As you see, it's a big protein. It's a glycoprotein. It spans the cell membrane. It has got three parts: extracellular domain protein, intramembrane domain. Here you see the chain spans the membrane 7 times. Therefore, it is called a 7-span receptor. They have another name for this, G protein coupled receptor, GPCR. Professor Rita Singh will give more detail. And the third part of the same protein is the intracellular domain, the cytoplasmic side. As you see, the carboxyl end is on that side. So it starts with the extracellular domain, the amino terminal. You understand a protein has 2 terminals. Professor Yogendra Singh has told you the detail, amino terminal and carboxy terminal. The amino terminal of this membrane receptor is on the extracellular domain. The carboxy terminal is on the inside or cytoplasmic. Most of the membrane receptors look like this with changes in chemistry. And then people asked, why should I measure this receptor or how do I measure? There are different techniques to do that. For want of time, I won't talk about it. There is a technique where you can take this source of receptor, incubate with increasing quantities of the hormone, labeled hormone, then separate the bound hormone from the free hormone and by filtration, and measure the bound amount of hormone because you measure radioactivity, and then do what is known as Scattered plot, as it is given here. B by F you plot against bound in terms of femtomoles. You get this curve, straight curve. The slope of this will tell you affinity of the receptor to the hormone. It is called KD. As you see here, it's about 3.3 into 10 to the power of 9 minus 9, which simply means it's an extremely high affinity. Similarly, it is also shown in this figure that if you take normal animals and those animals which are so-called fasted for some time, you notice that the number of receptors to thyroxine or triiodothyronine decreases as it is shown here, control and fasted. That means receptor number changes during physiological changes. That's why it is important. Similar result I have shown here for HCG, another hormone, binding to its receptors, and you compare the control animals and those which are diabetic and those which are diabetic and given an injection of insulin, you notice that the receptor number changes. When you become diabetic, the receptor number decreases. That's how severe diabetic people cannot reproduce. This is a reproductive hormone. But if you take insulin, it is restored partially. Okay. Therefore, modulation of receptor number and affinity is one way of regulating physiological activity or tissue response to the hormone or drug. So this conversion of this presence of extracellular signal after binding to the receptor into some changes in the intracellular, it goes through what we call as cell signaling pathways. She will give you more details about it. I'm only summarizing. And therefore, a variety of signals from extracellular metrics, from other cells, nutrients, hormones, environmental pollutants, they or attack this particular target cells, and then the tissues or target cells respond to that. What kind of responses you have? There are two types. Metabolic responses, which means they happen in minutes to less than 1 hour. So if you take, for example, insulin, blood glucose levels fall within half an hour. Similarly, if you inject FSH, you can see glycogen concentration increasing. This is a metabolic effect because if you add cyclohexamide, it does not have any effect, which means protein synthesis is not involved. However, there are other hormonal effects which are called genomic effects, which means there are alterations in gene expression which will take hours, days, and weeks. When an animal grows in response to growth hormone, tremendous amount of changes take place in gene expression. And that genomic effects are sensitive to both actinomycin D, which is an inhibitor of transcription, as well as cyclohexamide, which is an inhibitor of eukaryotic protein synthesis. So there are two types. So in a variety of fields, in endocrinology, in developmental biology, in immunology, in neurobiology, you see these responding receptors, and through cell signaling, there is a lot of alteration in cellular target cell metabolism, which gets translated as physiological effect or developmental effect or behavioral effect. Same type of mechanism. And therefore, if you summarize now, receptors exhibit three functions. Signal discrimination, they are able to bind a particular hormone even though concentration of other similar structures may be there in gram quantities in the blood. Therefore, they are able to discriminate signal from noise. Signal transduction, how this is carried across the cell membrane, and signal amplification through second messengers and all. She will give you more detail, and that is the summary of hormone action. Now we should say tissue response to hormones and not hormone action. Hormones don't act. And these second messengers, because the hormone is not entering the cell, it will tap the door and somebody else will take the message, and that is called a second messenger. In history, you will see half a dozen second messengers were discovered. Some of them got Nobel Prize. Cyclic AMP was the first second messenger discovered by Sutherland. For want of time, I'm not going into detail. It was a serendipitous discovery. And then subsequently, people discovered other second messengers like inositol triphosphate, adenosine, cyclic GMP, nitric oxide, and many more gases and chemicals. And these then, through some mechanism, amplify this signal, which is characteristic of hormone action. And that is the third part of the receptor function. And we'll take one example and show how nicely this is done by a simple post-translational modification, which is phosphorylation. So regulation of blood glucose, if you take as an example, many hormones are involved. We will discuss only the action of insulin, glucagon, and glucocorticoids with regard to this blood glucose. Now, long back, Most of you know what glycogen is. It is a storage form of energy in most of the animals. And this, you see that liver has a lot of glycogen. Every tissue has glycogen, but maximum store you will see in liver and muscle. There was a husband and wife team, Carl Corey and Gerta Corey. Long back, they discovered an enzyme called glycogen phosphorylase, which degrades glycogen, and the resulting compound is glucose 1-phosphate. If it happens in the liver has an enzyme called glucose phosphatase. Therefore, phosphate is removed, and the glucose goes across and then enters the blood. And therefore, liver glycogen contributes to blood glucose. And this is the summary of their work, their Nobel laureates. Long after that, when another postdoc joined their lab, Earl Sutherland, he was asked to explain how do they bring about this. He did a lot of work and discovered cyclic AMP. For that, he got Nobel Prize. Subsequently, similar work was shown in the case of neuronal cells by one of his students, Paul Greengard, and Greengard also got Nobel Prize. Subsequently, people like Martin Rodbell and Alfred Gilman did enormous cell biology work, gave us the details about what happens actually on the receptor molecule, and for that they also got Nobel Prize. Similarly, other people have contributed, especially Lutz Birnbaumer, Philip Cohen in Scotland, who was a professor of biochemistry at the University of Dundee. He contributed how this phosphorylation, dephosphorylation regulates the activity of phosphorylase B kinase and hence indirectly blood glucose. Phosphorylation of serine or tyrosine residues. This is a post-translational modification. It is catalyzed by a number of protein kinases. People have studied from microbes to mammals, plants, and discovered hundreds and thousands of kinases. We don't go into detail. Protein kinases and phosphoprotein phosphatases, the enzymes which will remove the phosphate. There are serine phosphatases, tyrosine phosphatases, hundreds of them, and they catalyze this phosphatase dephosphorylation and of the target enzyme or target protein. And therefore, during cell signaling, protein kinases and phosphoprotein phosphatases together regulate the activity. And this, in the case of glycogen degradation, results in decrease or increase of blood glucose. If you take this example like glucagon or epinephrine, when you give an injection of glucagon or epinephrine, it goes and binds to its cognate receptor. There is a second messenger released, which is cyclic AMP. And this in further activity regulates the activity of another one called cyclic AMP dependent protein kinase. And then that acts on glycogen phosphorylase B kinase. And then it becomes phosphorylase A, which degrades glycogen, and you get ultimately blood glucose. That is the summary of what happens when you take an injection of glucagon or epinephrine. I will take another 5-10 minutes to summarize what happens at the receptor level. This is all the work of Gilman and Rodbell. So the hormone glucagon in this case comes to the target tissue and then binds to that receptor. As is shown in this figure, the receptor has 3 proteins. One, the binding protein labeled as R, which is a biochemical receptor binding protein. There is another protein here, AC, adenylate cyclase. This is an enzyme. And there is another protein called G protein, which itself has 3 subunits, alpha, beta, and gamma. But what you notice here is that the 3 proteins are far apart in the cell membrane. When the hormone binds to R on the outside of the cell, extracellular side, something happens in the membrane. The receptor now bound to the hormone binds to the G protein. Now the G protein, as you see here, has 3 subunits, and one of them, alpha subunit, essentially has a bound GDP. But when hormone action starts, GTP gets bound to this G alpha protein. As a consequence, G beta and gamma dissociate. G alpha goes and binds to the binding protein, and together they go and bind to the adenylate cyclase, which means there is a diffusion of this ternary complex. And when that happens, adenylate cyclase is activated, and that faces inside also, cytoplasmic side. It's a transmembrane protein, and that results in the conversion of ATP to cyclic AMP. How this hormone action results in the activation, but we also know that when you take an injection, you will not get perpetual hormone action. Somewhere it stops. How does it stop? And that is shown in the membrane at the receptor level itself. When the hormone binds and activates G protein and through that the adenylate cyclase, I said that GTP is bound to the alpha subunit. But this G alpha protein is also an occult enzyme. It is a hydrolytic enzyme called GTPase. So immediately after few seconds, it will activate and therefore hydrolyze the bound GTP, and therefore the bound GTP becomes GDP, and GDP bound to alpha is no more active, and therefore hormone signal stops. So this is a mechanism of hormone inaction, which is a cycle. When you take an injection of a hormone, it acts for some time and then signal stops. This one mechanism in the membrane tells you how this stops. Gilman did this work and got a Nobel Prize for this. And then this activation resulting in cyclic AMP goes and binds to a protein. This is what was done by Edwin Krebs. He got a Nobel Prize for this. But I must inform you people that protein kinase was discovered in India 30 years before Edwin Krebs by a group in Madras University, Srinivasan and P.S. Sharma. They published on casein protein kinase, but nobody knew the importance of that. 30 years later, Krebs discovered this protein kinase, but that is a cyclic AMP-dependent protein kinase. For that, he got a Nobel Prize. This cyclic AMP-dependent protein kinase gets activated, and it, in its turn, goes and binds and activates phosphorylase B kinase. And that phosphorylase B kinase, as is shown here, it has got two sites for phosphorylation, alpha site, beta site. When the cyclic AMP-dependent protein kinase acts on the phosphorylase B kinase, it undergoes phosphorylation at the beta site. Once it becomes phosphorylated, here protein phosphorylase B kinase is active. What do you mean by active? It now phosphorylates another substrate or protein, which is phosphorylase B. But meanwhile, again, there is a regulation here. As long as the beta site is phosphorylated, this is a good enzyme. But moment beta is phosphorylated, subsequently the alpha site is also phosphorylated by the same enzyme. However, phosphorylation at the alpha site destabilizes the phosphate at the beta site, and a phosphatase comes and removes that. And therefore, you get back to a form of phosphorylase B kinase where only phosphate is present on the alpha site. And subsequently, another phosphatase will remove that phosphate, and you get the original enzyme, which is inactive. And therefore, a cycle of phosphorylation, dephosphorylation regulates the expression of enzymatic activity. And that's how regulation of glycogen degradation takes place. That's how blood glucose levels goes up or down based on the regulation of these enzymes. And therefore, this will tell in summary how one post-translational modification like phosphorylation and the opposite of that, which is removal of phosphate or dephosphorylation by an enzyme, regulates the amount of enzymatic activity and ultimately leads to changes in blood glucose levels. The other effects of hormone, genomic effects, I said they are sensitive to actinomycin D and cycloheximide. I'll give you only one example. This is an example where luteinizing hormone increases the length of polyadenylic acid tail of the messenger RNA in the gonadal tissue, specifically in the ovary. This was the first time reported by our lab long back. And We developed a new technique to measure the length of poly A by a very novel technique. This is another example of thyroxine action. All these are essentially genomic action. I will not go into it. I mentioned about the tadpole becoming a frog. This is amphibian metamorphosis. This is a genomic action. And therefore, people said feedback loops shape cellular signals in space and time. Summary of developmental biology. I will not go into the third problem. There are a lot of unsolved problems in endocrinology. Those of you who are excited can take this for your research career. Again, I will not go into detail. Suffice it to say what I have done. I have taken one example of a post-translational modification, continued Professor Yogendra Singh's lecture and showed how it is important in regulation of blood glucose. In that context, we summarized what are hormones, what are receptors, and how they bring about these changes. And it will also introduce you to the concept of receptor so that the next lecturer, next speaker will give you details of gonadotropin interaction with its cognate receptor. I hope I was clear to all the audience. Thank you very much for your patience.
Dear participants, you can post the questions in chat box. As there is a lot of disturbance, if you unmute the people, it is better if you can post the questions in chat box. Sir, one quick question. Sir, can you stop sharing the screen?
Yeah, I'll do that. Yeah. Tell me, sir. Hello?
Most of the animals you have discussed in this talk were mammals.
Yeah.
What about the other animals, lower animals?
Yeah, plenty.
Where they don't have much of the heart. They don't have much of the organs or organ systems.
No. Wait. Okay. Hormones are present in all the phylogenetic groups. However, what we are referring to is the major part was done on mammals, mouse, rat, elephant, and all that. However, there is a group of endocrinologists, they're called as comparative endocrinologists. The same thing you see in comparative biochemistry, comparative physiology, and all that, where people work on non-mammalian vertebrates like amphibians or reptiles or fish and have discovered the same thing. Slight differences are there, same hormones, same hormone action and all that. Not only that, you go down even further, invertebrates like cockroach or earthworm or mollusk, they also have hormones and there also a lot of work is there. Because there is not very significant difference between this and what happens in mammals, historically mammalian endocrinology was discovered first and explained. Therefore, I focused on that. But the same thing happened in other things. Not only that, botany people discovered hormones, hormone receptors, and all this GPCR and all 30 years after the animal fellows did. So there is a plant endocrinology also. We don't call this, use the word endocrinology there. You know the adenyl the nucleotide derivatives, they're all growth hormone there also. You have ethylene, which is a fruit ripening hormone. Like that, there are many hormones in plant system, but there's a much more mystery there because it does not follow, there is no blood circulation. So the movement is through diffusion. So all these differences are there. I will not go into detail, but please remember unicellular organisms, multicellular organisms, plants, animals, everybody has hormone. The name is different in different systems. That's all. This is all called as communication signal field, signal physiology. So if you use the word signal, it takes care of the entire spectrum of living organisms. If you talk hormone, we must talk about mammalian system, then non-mammalian vertebrates, and then invertebrates, and then plant systems. Is that answer?
Yes, thank you, sir. Now it's time for our next speaker, Professor Rita Singh, ma'am, to start her presentation. Ma'am, ma'am, you are unmute now. Ma'am.
Yes, good afternoon. Good morning. I think it's still morning.
Yeah.
Good morning, sir.
Ma'am, for a while, I'll make you a host. You can share the screen at that moment.
Okay.
Later on, you make me a host back. Because it is making a disturbance to others.
Okay. Can you hear me properly?
Yes, you're audible.
Okay.
I'm changing the host now.
Okay.
Now you can share the screen.
Okay. Can you see my screen?
Yes.
Okay. Can you see the slideshow?
Yes.
Okay. When do you want me to start?
You can while.
Okay. So shall I start?
1 second. 1 second. Good morning. We have today with us Professor Rita Singh ma'am, professor in University of Delhi. Her research interests are FSH, FSHR responses, high LH and bias signaling of FSHR, cell signaling in granulosa cells, molecular defects in polycystic ovary syndrome, susceptibility genes, lifestyle and environment. To her credit, the awards are French Visiting Researcher Award from Scientific Council, France, Professor L.S. Ramaswamy Memorial Award and Gold Medal in 2019 by ISSRF, Academic Visitor Award Yong Lo Lin School of Medicine, National University of Singapore. International grants for conference speeches and visiting scientists from France, Italy, Iran, Qatar. DBT International Award for Advancement of Science in USA. CREST CREST Program. DBT Indo-US CRHR Grant. DST Young Scientist Award. Member 9th Antarctic Expedition of India. And to her credit, the publications are plenty in high impact peer review papers, reviews, and book chapters. With these few words of introduction of the resource person, I welcome Professor Rita Singh ma'am to start her lecture and address the gathering. Ma'am, unmute yourself.
Yes, yes. I thank you, Dr. Khan, and all my gratitude to you and Professor Murlidhar for organizing such a, focused and very, very important workshop. And I want to thank all the audience and faculty members who are here. Please feel free to ask questions anytime in whenever this is in progress, because I'm going to take you through a lot of things, and some will be very simple and will go a little fast, but some will be very interesting because my overall area of research is or interest is biomedical and linked to the human interests in the whole venture of science. So let us start with um, the um, uh, first and the foremost thing, as my title said, that the glycoprotein hormones, which I am going to focus on FSH, are very, very important hormones, and they are every day with us for maintaining our health. So we will go step by step to understand what are these hormones. And because this is our academic lecture, so I will go through certain very essential informative things, and then we will also see how, if these things don't function properly, then what happens to our fertility or otherwise our health. Because these glycoprotein hormones or gonadotropins, they are not just the reproductive hormones, they have several dimensions. So today we are going to talk a lot of dimensions also about the physiology of these hormones. So in the first slide, I'm just showing you that these inside the ovary, there are a number of type of cells which maintain our fertility. The most important one is the oocyte, but there are other nursing cells which are called granulosa cells. Now, these cells do a lot of churning of metabolites and signaling molecules and hormones, and so that oocyte does not have to really sweat out. So it is very, important connection between these two. In fact, they have a physical connection between these two type of cells, and lot of signaling molecules, metabolites, and several other things, they are communicated through this. Some of the important metabolites which are known to be regulated by FSH are pyruvate, lactate, but now we also have in this list glycogen, and this is independent of the insulin. So FSH has independent action on glucose metabolism. So these things we will go through in little while. So FSH binds to the FSH receptors on these granulosa cells. This is the specific thing which happens in females. But in case of males, earlier people thought that FSH is a female hormone because the name goes follicle stimulating hormone. But now we have realized that it is one of the important regulators of spermatogenesis also. So FSH binds to Sertoli cells, and it is very important for spermatocyte maturation. It also is important for various kind of feedback mechanisms in the males. So this has a lot of important dimensions which we will see later. But first, an important story. Now these glycoprotein hormones, FSH and LH, these were discovered by a scientist called Hisaw. He was a professor of zoology in University of Wisconsin and later in Harvard. Now this scientist, along with few of his associates, Favold, who was a chemist, Leonard, who was a graduate student in this work, they all did very, today we will call them very simple studies, but they just put the pituitary extract in immature rats, and they found that ovarian follicular development was there, and there was also precocious sexual maturity in immature rats. And later on, they also discovered LH. Although given alone, it did not have much effect on the follicles, but once it was given with the, a particular combination or the concentration, it caused the follicles to luteinize. So this was a seminal work by Hisaw, and how this was these gonadotropins were discovered. Because early in the history, they were doing these substitution experiments. They will make extract and they will put in immature, or they will do surgery and remove the endocrine organ, and then they will inject into that, and they will find that there's something going on here. So the things went on. But slowly in the history, the glycoprotein hormone family has expanded. It has expanded to contain a lot of similar looking biochemical protein or glycoprotein hormones. I have told you about FSH, and this is made in anterior pituitary gland. And obviously, we know mammalian reproduction, or it is very important. But how it functions is the interesting thing which has come up till today. And this is because of its binding to its specific receptor, which is called FSH receptor. And this has so these gonadotropin receptors have co-evolved due to gene duplication events. Now the other hormone, gonadotropin, is LH. But another hormone, which is the placental hormone or the chorionic gonadotropin, functions like LH. Most of the time in the experimental biology, when we are doing research in the lab, we did not have access to LH because recombinant LH has just come recently. But HCG was available because of its isolation from the urine of the pregnant women and like that. So now we have all kind of recombinant hormones, but these two hormones were earlier thought to be same. But recent studies by certain scientists in Italy, Casarini, I know him very well. Now they have compared the activity of these two hormones, and it is like HCG has some 5-fold more activity than LH. And then in this member of this family is another hormone, which is thyroid stimulating hormone, which is basically maintains the overall physiology and does a lot of cooperative responses. And it is a non-gonadotropin, but this comes in the glycoprotein hormone family. LH, as I said in the last slide also, that it is important for the ovulation and for luteinization, formation of corpus luteum, which produces progesterone. It is also very important for the fertility because conception will happen when there is proper luteinization. LH is also important in males because it is the most important regulator of spermatogenesis and steroidogenesis, that is production of testosterone and other androgens. So all these hormones are produced by specific cells in the pituitary, gonadotropes, thyrotropes, and other hormones you can see here. And they belong to the anterior pituitary hormones family. So here these hormones, though they are in the same category, but they may have different molecular weights. But interestingly, when we go to their structure, we have, I will once go to the slide, this one. So most of these hormones, including HCG, I have not written here, they have two subunits, alpha and beta subunits, and these have certain disulfide bonds. Now, all these hormones of the glycoprotein family have the same structure, but then their specificity of responses are maintained by their beta subunit. So we will see that how microheterogeneity of beta subunit can also give rise to, today in the advanced biology of FSH, I'm going to explain that also to you, something called, differences in the responses of microheterogeneous forms of hormones and receptors. So we go back to the structure of these hormones. So this is, we know it's a glycoprotein. It is has two subunit. Alpha subunit is 92 amino acids and the beta subunit is 111 amino acids. So the specificity of this is very important. But another important thing is that these subunits are glycosylated, and this glycosylation is very important for the half-life of these hormones. So please note the time of the half-life. So FSH is there in the blood for its half-life is 3 to 4 hours. It means that its concentration will go to half after every 3 to 4 hours. Then comes LH. Now, interestingly, it also has both the subunits. Alpha is 92, beta is 120, and its half-life is only 20 minutes. So you see the difference in the half-life. So you will realize how the microheterogeneity is another thing, but the overall balance of their structure is so different. Now comes the HCG, which is similar like LH in its responses. But if you see, there is difference in the glycosylation. It has many more glycosylation moieties on this, which, comprises 30% of the molecular weight of HCG. So it is more. But then you see the half-life is almost tenfold more than FSH or many-fold more than LH. So, so if you see what changes this half-life. So one of the important thing which changes the half-life of these hormone is the glycosyl moieties or the glycosylations. That means the extent of glycosylation is the one which is going to govern how much time these hormones can stay in the circulation. So let us see the glycosylations. So if you see in bovine, ovine, porcine, equine, I hope all the students understand these words. Bovine means the buffalo cows, ovine means sheep, porcine means pig, equine means horse. So these have glycosylations. Here you can see the amino acid number. In human, it is slightly different. If you see, it is 52 instead of 56, and it is 78 instead of 82. And the kind of glycosylations, you can see they are either the serine threonine linked or asparagine linked. So now when this alpha subunit is uncombined, because blood will have some uncombined alpha subunit, that also has been linked to the history of the, some defects in patients. So, so if you now go to the beta subunit, bovine, ovine, and porcine have one glycosyl moiety, and then human also has one, but you look at the number of amino acid where it is. Equine has same as these, but it also has additional serine threonine glycosylation. So you see there is certain amount of variation in the glycosyl moieties, number of them, and where they are located. So let us look at FSH. It is very interesting. So now if you see in bovine, ovine, porcine, equine, human, FSH structure is same. So this is very interesting that this is the hormone which has many roles in the physiology. So let us look at how the glycosylated heterodimer of 2 subunits binds to the receptor. This is extracellular domain of FSH receptor. And this is alpha and beta subunit. You see there are these glycan, it shows the glycosyl moieties, and the receptor also has certain amount of modifications. So this gives rise to specific response. So now before we go on to all those things in more details, one important thing I want to make here to bring your interest in this field, this particular hormone, which we earlier called follicle stimulating hormone, has gone beyond fertility. So how this FSH has role beyond fertility is something you will find. One example to excite you, I will give, it has a role in cancer also. So let us go through this whole lot of information and then try to understand how this hormone plays a role in gynecological cancers. But before we do this, we must spend some time with HCG. HCG, as I told you, is also like LH, and it also has alpha and beta subunit with more glycosylations, you can see. And you know, these both alpha and beta subunit, they are found on 2 different chromosomes. So you see, and here you can see the sequence, how these you can see these round things here, these are the O glycosylations and N glycosylation in the structure. And these glycosyl moieties are very complex structures where you have various other like N-acetyl glucosamine, galactosamine, mannose, like and sialic acid at the top. And in my PhD, I worked on another important modification, which is sulfate. Sulfate also is there on the blocking of the glycosyl moieties, and it also is modulating the half-life of these hormones. Many of these hormone levels are, measured in our diagnostic labs by ELISA. And these sandwich ELISAs or competitive ELISAs, these are direct, indirect, many kind of ELISAs are there. So these, I'm not going to go into detail, but this is one of the technique you must know that is important for studying the level of hormones in blood. So using these various technologies, it was really amazing to find that certain non-gynecological tumors also are positive for beta HCG. Lung carcinoma. So like this, if you see, these gonadotropins are linked to cancers. But one interesting thing, I come back to glycosylations again. The hyperglycosylated HCG is correlated with cancer. Cancer invasion, the choriocarcinoma invasion is enhanced because of hyperglycosylated HCG. So if the glycosylation becomes defective, whether it is hypo or hyper, I'm giving one example, even hypo becomes, in certain ways it causes the, the gestational diseases. But in view of the cancer and related, etiologies, this is a virtual absolute marker of ongoing invasion or malignancy. So now hormone, which is HCG, which is made naturally from the placenta, sometimes starts getting expressed from pituitary as well. Sometimes post-menopausal women may have these defects, but if it is the normal level in premenopausal, maybe 5 IU per liter or negative maybe, but post-menopausal may have little higher. If you understand menopause is at the age of 40 to 50 years of age, females stop their menstruation or periods, and then after that, estrogen is not formed, so the gonadotropins increase in their blood. FSH, LH levels will increase, but Here again, pituitary sometimes defective way starts making HCG, which can also be not good for the body because it can be linked to cancer. So therefore, the hormone replacement therapy has to be done, or the oral contraceptives have to be taken to suppress this, and maybe then it might not just trouble you more. So if we now come back to a little bit of academics, so this is very interesting. This lecture I gave for my MSc when Dr. Murlidhar was my teacher, but I made that time on transparencies. We did not have this luxury of 2 minutes, you make a slide. So I had to draw on all these, hexagonal boxes I drew on the transparency, and then um, I showed. So these are the structures which get, N block transferred on the amino acids. So this is something which I want to share because it's very interesting. So these N and O linked oligosaccharides, how do they get to the protein chain? This happens in the endoplasmic reticulum and the Golgi complex area. So now in the endoplasmic reticulum, when the protein is being synthesized, something which gets into the protein chain while protein synthesis, we say it is co-translational, or if after the protein has synthesized, we say it post-translational modification. So these things are happening in endoplasmic reticulum and Golgi. So then from there they get into the other their target locations. But the important point which I want to make regarding the glycosylation is during their process, after their N block transfer to the protein, these proteins pass through the Golgi, and this is the portion where these glycosyl moieties, which were maybe simple in nature, they get more complex in terms of addition of sialic acid or sulfate or many other modifications. From these simple mannose rich chains, they become more complex with N-acetyl galactosamine or glucosamine. So those things keep adding on during their visit to Golgi apparatus. And after the complexity is built, so this is the very interesting thing which I felt during my MSc days, that this dolicol phosphate is the one which builds these glycosyl moieties, and then from dolicol they are N block, transferred to the amino acid. So this picture shows you how these are formed, and then these can vary from you can see that different colors show different type of glucose moiety. So the complexity is built up, and so these things are highly regulated. So there are several enzymes, and those enzymes are regulated. So that's why HCG is different when it comes to glycosylations. LH is different. FSH is different from within the same family of glycoprotein hormones. So just to tell you what this dolicol is, which is acts like a father and puts the glycosyl moiety there. This is nothing but has the origin like cholesterol, and then these isoprenoid units, they are put together. And interesting point, which that time also Dr. Dureswamy asked me the question and I knew the answer, so till today I remember it. So this whole process of building up of these glycosyl moiety is energized by not ATP but by CTP. So this is very interesting that a specific energy donor is different here in this whole process. So this is dolicol. This is a simple isoprenoid structure. So now we come back to the story of FSH, FSHR, signaling, and other interesting aspects. So now FSHR earlier was thought to be expressed on ovarian granulosa cells specifically. But now we know that FSHR is also present on ovarian epithelium. And that's why it is also linked to ovarian cancer. Ovarian epithelium has certain, stem cells, and this is the one which gets converted to the carcinogenic tissue. So that's why FSHR is starting to get its, um, importance as a marker of cancer. Let us see what is happening with this particular receptor and how this is getting into this role. This was expected because we know if we give FSH, whole of folliculogenesis happens. How? It can do proliferation, it can do steroidogenesis. We know it converts testosterone to estrogen by stimulating CYP19. So all these various different functions, including the metabolism, is taken care by this hormone. So, like if we multiple responses by a hormone is nothing new, but then getting into a role which was known earlier to this new role came because of the differential expression of FSH receptor. So now this receptor is also found in vascular endothelial cells. Now vascular endothelial cells are very important, as you know, because angiogenesis is possible because these cells grow, they make vessels. So these vessels are FSHR positive. So when during tumorigenesis, angiogenesis happens, or in the ovary, angiogenesis happens, so the role of FSHR in angiogenesis or even neoangiogenesis may come up in a larger way, and also in tumor metastasis. But if we look at this receptor, we tend to believe that FSH can do so many things because this receptor itself is a complex structure. It is a GPCR basically. We have so many GPCRs in our body. Adrenergic receptor is a GPCR. Growth factor receptors are also EGF receptor. Many other receptors, they have either properties like GPCR, or they do something called trans activation. They get trans activated. So those things I will not go into that, but certain amount of it I will tell you with examples. So this FSHR, I'll go to the structure here. So if you see, FSHR is a 7 transmembrane receptor. It has these intracellular loops, you see here, and a smaller C-terminal extension. But if you look at the extracellular domain of FSHR, it's a large domain. And this, when it binds to FSH here, how it can give rise to so many responses? This is the question which we are trying to answer. First, we have figured out that it can do so many things, but now the question is how. So let us look at certain things and try to understand what it's doing. This is another figure just to show you that how this whole thing looks like. This is extracellular domain, and these are transmembrane, a simplified version so that students can understand. So this is the plasma membrane, this is the receptor which is there inside, and the large extracellular domain then binds the FSH. You see this movie, it activates adenylate cyclase through GS-alpha, then cyclic AMP and PKA. This pathway takes care of follicle growth, granulosa cell proliferation, germ cell survival, proliferation, differentiation, steroidogenesis, metabolism. But if we go back again and see the movie again, it is just that, wait a second. Okay, so now this is not just the only thing which is happening. You have other receptors which are insulin receptor and IGF-1 receptor, that is insulin growth factor receptor, which synergizes. We have seen these papers over the years, but the important thing which from my lab, that These receptors are dependent on FSH and FSH receptor signaling to induce the expression of insulin receptor substrate 1 and 2. So these receptors are very important downstream of insulin receptor and IGF receptor to activate PI3 kinase and AKT. And this now AKT can do various kind of responses, which you see if we knock out this IRS2, you see what happens. IRS2 knockout mice are infertile. And the interesting point when we started this work was that these mice were resistant to the responses of gonadotropins. So these were the important studies which induced in our mind that there is something going on here. So it means if these IRS2 knockout mice are resistant to gonadotropins, it means that these are important for gonadotropin action. And that is what we discovered in our lab and we published. So this is here, you see. So this whole pathway, which is AKT, the survival factor, means it is very important for cell survival overall in the whole of cell biology. But let us go slowly further and try to understand it slowly. So now this FSH receptor, what it does, it binds for, slide is for students and the newcomers in this field. So it binds to receptor, but after some time it gets desensitized. But interesting point is FSH receptor is not like other receptors. We are still trying to understand its physiology, but finally it gets desensitized. And then this desensitized receptor can go to various fates. Number one, it can either get into a pathway, which is, the pathway where this whole complex will get recycled to the membrane. It will go through the endosomes and go to the cell surface again. But it can also go through from endosomes to lysosomes, where it will get degraded. But most of the time, keeping the balanced energy view, it gets recycled. So here are the important kinases which are called GRKs. Now these, they phosphorylate this FSH receptor where beta arrestin binds. And then this whole process of endocytosis happens and this hormone receptor bound complex get further metabolized. But the important point for you to understand how this FSH stimulates FSHR. It's a very interesting story. Now this is a FSH receptor, okay, the pink one, and this is transmembrane. Now comes the glycoprotein hormone, that is FSH. The purple binds to the high affinity binding sites here. This complex alpha beta binds to the high affinity site first. Then it makes a confirmation that this particular residue of the FSH then interacts with this sulfated tyrosine at 335 position of the receptor. And once this binding happens, then the receptor is activated. So based on these interactions, various mutants are formed, and then their relative activity has been studied by various scientists. I will show you how this binding studies with mutants and other things have brought out a new concept of therapeutics. So if you see these receptors, they not only make activation through single receptor, as I thought as a student, but these receptors make either dimers or oligomers, even GPCRs. These are big molecules. If you see, their molecular weight is high. So still they are making dimers or oligomers also. I'll show you some pictures. And then they have various motifs. If you see here in this, you see it has certain glycosylations here on the N-terminal portion, but at the C-terminal, it has several motifs. Now these motifs, if you see, if you have knack for this biochemistry of these structures, you will see that these different motifs are very important for various kind of activities of these receptors, that is the receptor activation or the internalization or various kind of activities which it is doing. I don't have that much time, would require I can give a detailed uh, uh, this thing. But what importantly you must understand is that these FSHRs, they make dimers which are called homodimers, or they can also make heterodimers. Something which was proposed and we showed in our lab and we published last year, that LHR and FSHR, they dimerize, and this dimerization is something phenomenal. Means that depending on the hormone levels, we showed that depending on the level of LH, I will show you certain data and I will explain you this aspect of these dimerization. And these oligodimerizations oligomerization can also happen, in which you see it is a kind of 3 structures of FSHR and 1 of LHR. So these are various proposals. These are not yet proved, but we have clearly proved that they make dimer and it affects the physiological response. So both the things we have shown in our lab. So I will show you that little bit of data. But before that, you must understand that this whole stages of folliculogenesis, starting from primordial till the Graafian follicle before ovulation, it takes 3 to 4 months for this primordial follicle to reach this stage for ovulation. So during this process, so many follicles are seeing different hormonal combinations and different LHR, FSHR expression combinations. God only knows how it is maintained and why they made it like that. But then that was the only choice now we are trying to understand. Because if you want so many responses to be generated, you want to, push certain kind of, mixes and balances that the whole set of responses remain specific and non-redundant. So this is what happens. So depending on the level of FSH and FSHR, LH and LHR, LHCGR means that this receptor binds both HCG and LH. The responses generated can be either cyclic AMP production. If it is heterodimerization, it may be biased towards calcium signaling. So, but then your G proteins will be different. Here, calcium signaling is stimulated by G alpha Q, whereas G alpha S is stimulating vinylate cyclase and cyclic AMP formation. So this aspect of, FSH and FSHR binding has been, made more interesting by various kind of defects which we see in fertility, level of defects which we see in fertility or in the reproductive cycle of women. So various kind of mutations have been discovered in the receptor and which can affect the FSH binding to the receptor, or some which are redundant, they don't affect the binding of FSH to FSH receptor. So there is only one mutation which has been proved to have certain type of effect on the FSHR and FSH binding and responses. So to understand more about FSH and how its role is coming in cancer more strongly, let us go through certain other things. So now, as I mentioned, that FSH and LH have their role in metabolism. You see this beautiful picture shows you that these are the granulosa cells and the staining is done for GLUT4 protein. So when you give FSH, GLUT4 protein goes to the cell surface. These granulosa cells are normally hexagonal and not very round cells. These have different kind of shape. So you can see that these cells your GLUT4 has gone to the surface. So FSH we showed, and this paper also is there. So FSH stimulates GLUT4, then glucose uptake is there. I'll show you in a summary slide. But when you look at FSH and HCG, it's between the glucose uptake, concentration dependent. HCG has much lower EC50 value than FSH. Higher EC50 value than FSH. FSH is more efficient in doing glucose uptake. Whereas we found, I don't think I've included that data, LH is more glycolytic. So it will utilize the energy or glucose more, whereas FSH is storing the energy, which we showed that FSH regulates glycogen synthesis directly independent of insulin. You see this beautiful, now this is a cell culture technology where we have not added insulin, we have not added any serum. Wait. So you see the glycogen granules can easily be seen. So this function of FSH independent of insulin was amazing to us, that it is just behaving like insulin. Rather, we found that it is much more efficient than, the combination of hormones which we have. So in summary, if you see, FSH binds to its receptor, activates cyclic AMP PK pathway. Now it activates a transcription factor with SP1, which goes to and binds to IRS2 promoter. Then IRS2 is synthesized. This is now available to insulin receptor and IGF-1 receptor, which when activated, they cause the intake of glucose inside the granulosa cells. So all this we have shown and published. So this is very interesting that FSH independent of insulin can do the glucose metabolic pathway. Why we are saying insulin independent? Because it is doing glycogen synthesis. But then before I go on to tell you that, you must know that all this we have understood by studying PCOS granulosa cells from human patients. So in these patients, GnRH pulse is defective, LH is high. So the ratio of FSH and LH is not good. So we hypothesized that LH is causing defect in FSH signaling. So when we went in that path, we discovered what I showed you earlier, but we published that and we showed how LH can cause the defect in this whole process of PCOS, so as that these women with PCOS are more prone to develop metabolic disorders like insulin resistance, obesity, dyslipidemia, hypertension. So all these things are happening in PCOS, and people were wondering, they did androgen trials in diabetic men also, but it was not androgens which were causing the diabetes or the obesity or things like that. So they also tried with estrogen, but then it was discovered that basically testosterone gets converted to estrogen, and estrogen may have certain insulin sensitizing kind of responses, but that also cannot be completely correlated, because the results were not reproducible, and depending on various situations, the role of estrogen could not be ascertained. But in our study on granulosa cells in culture, it was very clear that the FSH and HCG, they both do glucose uptake. FSH causes glycogen synthesis. Here I'm not having that data. Let me see. No, I have not included that data here, but my paper I will show you. You can deal with that. And but what we found was that FSH increases glycogen synthesis, so but LH causes glycolytic pathway activation. But in case you have high HCG or imitating high LH in PCOS, now this function of FSH was defective, means LH was interfering with this. And we did concentration dependence of HCG in FSH incubations, and we saw it is causing absolute inhibition of FSH stimulated glucose uptake. So we propose that LH is reducing the FSH sensitivity. Now when we check the literature, there was, HCG attenuates insulin sensitivity as well. And when we checked in granulosa cells, we found, yes, HCG is also, causing the insulin defect. Then high LH was also causing decrease in FSH sensitivity in vivo response, where it was not directly shown to be, through FSH receptor and all that. So when we did the experiments with granulosa cells, we clearly found, and the interesting point was that in most of the PCOS patients, what happens is that their steroidogenesis keeps going on, whereas the metabolic pathways are defective. Glucose uptake and glycogen synthesis, we discovered this, and it was very, very interesting. So we called it selective defect in FSH response in PCOS patients. And we have shown that in PCOS non-IR patients, when you add,
It's 12 o'clock.
HCG to these normal responsive females, there are females which have defect in non-IR. So there may be certain mutants of FSHR, because these women get FSH recombinant. So there is a defect in FSHR binding or, response in these women. So this was very interesting, that PCOS IR has a defect in FSH responsive glucose metabolism. So then we went ahead, and in vitro system, we tried to understand the interaction between these receptors. And so first time we did this co-transfection of, we have construct of LH receptor and FSH receptor. Now these are bound to these, Brett probes. Like these are certain, fluorescence probes, RLUC8, and this is yellow fluorescence protein. Now these are such that if these two will come up, it will fluoresce more. So there is certain kind of coordination here between these two probes, that only when they come close that one of them fluoresces more. So this is technology which can be measured with a specific, very sensitive machine, which is very expensive, and India does not have a single machine like that. So we are lucky to get an award. They are very happy that this kind of work can lead to a lot of therapeutics. So they have given me this award so that I can go there and they can facilitate this work, which was left in 2018. I had gone there. So we have a long-standing collaboration where we have very interestingly made this assay system. So now in this, we can check how we can interfere with this binding by a therapeutic peptide, so that if we can remove this defect of this heterodimerization, we'll be able to stop this, So when these hormones are alone, they will not cause the defect. Only when they are, together. This is my collaborator Pascal from France, and this is another, my colleague from US, and we all have a very active group on these studies. So, but when these hormones are together, they do magic. At very low concentration of HCG, we found very interesting thing, that it was synergizing for glucose uptake with FSH. But if you increase the concentration, it causes the inhibition of FSH responses. So we got a first time in the world for these hormones, we got this model system where we can easily study these aspects of receptor interactions, hormone interactions. These are some of the results from our PCOS patients. You can see that estrogen levels are either normal or more in PCOS patients. But if you see LH levels, they are more in PCOS patients. So high LH is not troubling the estrogen synthesis. Rather, it is now known that LH also stimulates CYP19. So it stimulates. So now we are studying whether this there is going a balance of higher estrogen levels than required along with higher LH. So these are the complexities where we are trying to understand the selective. So this is our recent paper. It's a big paper. You can go through this. And there's a lot of things to understand in this and more to understand with our going on research work. So what he said made us and many groups abroad, they were proposing this bias signaling in adrenergic receptors. But in FSH and LH receptors, we have proposed this based on our data. So what is happening here is biased signaling means that a receptor adopts multiple conformational states. It's very important for all of you to understand. One GPCR can take many conformational states. Now, this may be active, inactive, partially active, selectively active, that we have shown in our model system that it can be selective. So these different conformations of a GPCR, which can give rise to different or great responses or entirely different responses, depending on the engagement of downstream molecules, is termed as now the FSHR or any other lot of studies are there in adrenergic receptors, biased signaling. So now we want to use this knowledge for developing therapeutics for defects in these. So there is now another level of medicines which are being tested. And so in this, what is done is that small chemical ligands are being synthesized and planned for FSHR transmembrane domain. Now, if you modulate the transmembrane domain, it may change the conformation of the receptor outside, inside, and it may lead to a different conformation overall, giving rise to the kind of signaling you want. So that is how the small compounds are being checked for modulating the receptor ligand binding. Then this is happening, this bias signaling is happening as a consequence of mutation I mentioned earlier, and because of polymorphisms. So there may be certain amount of changes in the receptor. And these, if we can design these small peptide or chemical compounds which can interfere with these defects due to mutations and polymorphism and lead to the effectiveness of the FSHR and FSH responses, then we can remove many infertility, mental tortures with women have. Then there is obviously this conditional effectiveness. This is depending on the concentration of the, or like FSH, LH. So once we understand that, we know that if FSHR is all right, we have to maneuver the concentration of LH and FSHR. I will show you something very interesting in pregnancy and how this thing is coming up, very interestingly here. Though a scientist talked about it many years ago, nobody gave a thought about it. But now the role of FSHR is coming in pregnancy also. So these things are making this whole area of research very dynamic, multi-dimensional. So if you look at this, so now again, I come back to certain academic slides so that you understand what I'm saying. So if you see, this is a receptor. It cannot bind to this hormone. There is another receptor which can bind to but cannot give rise to response. So this is a signaling deficient mutant and this is a binding deficient mutant. So when they are co-expressed, the binding and the signaling deficient, this thing, so then what will happen? There will not be any through this receptor, but the binding of this receptor to this may activate this green, arrow shows the trans-activation pathway may just function. So you see, this heterodimerization can lead to a concept which I earlier just took the name trans-activation. Trans-activation means this receptor binding itself can cause trans-activation of certain signaling pathways. Otherwise, we know in adrenergic receptors, they have this cross, trans-activation through your, so and this is trans activation. So there are many aspects which are very interesting. I've discussed some in my paper, which I showed you in metabolism in the discussion section. So let us see this one to make you understand more. So now if this is FSH, okay, it is activated. So now this activates the G protein pathway, and it also activates the beta arrestin pathway, because this is going to get desensitized finally. So this is wild type receptor. So now this is another situation where now binding with heterodimerization with another receptor may just make it biased towards G protein pathway, and beta arrestin is not happening. So what will happen? This is going, cyclic AMP pathway will remain active. So this is a defect. As you know, cholera toxin activates G protein coupled receptor in the stomach, and you have diarrhea all the time because the receptor cannot get desensitized. But here, if you see, this situation may lead to this, and there can be another situation where it's heterodimerization with another receptor which is defective or mutated, may lead to over activation of beta arrestin pathway. So this will remain desensitized, or beta arrestin pathway may activate certain downstream pathways. Now recently there are some new reports that beta arrestins can also activate certain additional pathways. So this leads us to another situation which I mentioned briefly earlier, that elevated FSH levels are there in post-menopausal women and also associated with ovarian cancer. Let us try to understand this situation, that if elevated FSH levels are there and a person has normal FSHR, because post-menopausal means normal fertility women will have both situations. So now these elevated FSH levels, they can activate several pathways which can lead to cell proliferation, cell migration, cancer cell invasion through these various pathways. And then this can also is known to promote EMT, epithelial myoepithelial transformation. So this mesenchymal transformation, I'm working on those cells, so I always say that. So these are epithelial mesenchymal transformation. So this whole concept of, just having something normal does not exist when the situation becomes post-menopausal because of the high FSH and LH levels. And similarly, there can be increase in tumor angiogenesis, and we don't know what to do about it. And that's how it is important that we have certain therapeutics which can inhibit FSH action, which can modulate FSH, FSHR defects, and one can. So if you see nowadays, these are markers for cancer. So nowadays, this whole list of markers which are under the target for treatment is FSHR and LHR. So G protein coupled receptors, which are of, earlier called as reproductive hormone receptors or the glycoprotein hormone receptors, they are now under the camera for treatment or the cancer therapy. So this is because these can do a lot of, these functions which promote cancer, and that's why this is a promising tool for oncology, and one can design drugs. So these are the three type of drugs which people are trying to develop, not in our country yet, but in most of the Europe. Now these small molecules, they can, as I told you, not only remove the can be designed to remove the defects in the binding and signaling of the gonadotropin receptors, but they can also be, made in such a way that they can promote selective signaling. So wherever there is a defect, you remove the defect. Wherever there is more proliferation, like in case of cancer, then you can make certain antagonists of these receptors. And there are certain, small peptides which are called nanobodies. So these are not full antibodies, but these are certain specific peptides specific for designed for modulating these receptors, and these are called nanobodies. So these are specific for binding to a particular receptor, so that there is no cross reactivity with the other receptors, and various kind of defects can be modulated, or they can be used for therapeutics. And then we earlier in the lecture we uh, we discussed the how glycosylated or hyperglycosylated HCG can be cancerous, or FSH can be defective, or LH can cause problems not only in youth causing infertility, but also in menopausal stage causing health issues. And we are linking also to the energy metabolism and glucose metabolism. So that aspect I have not discussed, that there's some certain proposals which we have and we are studying I think I forgot to put that slide on the pregnant women. So I will just discuss here that in pregnant women, what has been known is that the FSH receptor is expressed in the placenta. And FSH receptor expression there is very important for maintaining, as we saw, angiogenesis and all, which is very important for the placentation. So now there have been studies in mice where they have shown that if you knock out the FSH receptor in the endometrium, then it can lead to certain kind of fetal defects. This is very interesting because we never thought that FSH receptor is important for pregnancy, but it excited us, because that is what we are proposing in PCOS women. So now what is happening in PCOS women, if FSH receptor is defective in ovary, it is also defective in endometrial or uterine spiral arteries, wherever it would be. We have to still discover all those things. Then what is happening? That may be a major cause of infertility in these women. We are treating them for anti-androgens. We are treating them with metformin and everything. But if FSHR is defective, that means it is going to be difficult for them to conceive. So we need these nanobodies or the modulating compounds or peptides to first cure or modulate their defective receptors. So depending on the extent of defect in the FSHR, one can, so we are very happy to have a model system where we can show that there is a selective defect in FSH receptor. And now we are going to work on therapeutics in this summer award period. I'm going to check certain nanobodies and peptides which they have, whether any one of them can modulate this, this heteromerization aspect. And if you can remove that, then it will be very good modulating peptide. So these are the important key points which I want to say at the end of my presentation, that FSH is not just a fertility hormone. It is important for complete health of a person. And all these exciting, observations in the literature, little bit we have contributed. A large amount of information has been contributed by Professor Muralidhar. So we are just have done minuscule of work, and we expect that we can do more, meaningful further work, and with his guidance all the time with us. So thank you very much, and please tell me if you have any question. These are various students of mine who work in my lab, and we have various collaborations in aims of Obs Gynae department. Then I have another collaborator who was the president of Fertility Society, Gauri Devi. She's a renowned surgeon. She was the only referral center when I started my research in 2002. And then this is my Indo-US award. He's a renowned person, Jerome Strauss. You'll see his books. He's a very renowned endocrinologist, and I was very privileged to have worked with him, or rather had him as my collaborator. So I just still, and we are still in touch. So it is very, very meaningful life, scientific, meaningful, work. So I have certain Indian collaborators and many of my students. So thank you very much. I have fundings from all funding agencies. CSIR is somewhere missing. So thank you very much, Dr. Khan, Dr. Murlidhar. My gratitude for giving me this chance.
Thank you very much, ma'am, for such a wonderful journey you have done with the hormones, and you have given us the insights on the hormones and its roles, and justifying the various ways how the hormones are going to be working out. So there is a question in a chat. You can see that in post-menopausal condition, will the hormone replacement therapy will work out?
Yes, definitely. I think women should not feel hesitant to take certain amount of, therapy for post-menopausal, health issues, because due to the absence of estrogen, or the, very, very low level of estrogen, FSH levels increase. So what happens is normal FSH level will keep giving you health benefits, but if FSH level increase a lot, it would be like, what we call insulin resistance. I am proposing post-menopausal FSH resistance. So if, FSH resistance is there, then FSH regulates bone mass. It also regulates many functions. As I told you, it maintains many tissues in the body. Maybe we are slowly working on what baseline functions FSH can have. One we have shown is the metabolism. That's why post-menopausal women, in the beginning, they don't have many metabolic problems, but once the FSH levels, they start increasing after 60s, then they start having many, because now this situation will be like, insulin resistance, where insulin is high and its functions are low. Similarly here, FSH levels become high, and it is most of its receptor non-gonadal will also get desensitized. So that is what we are proposing. So I think HRT is very, very important for post-menopausal women. There are certain safe ways of managing. It is not that you always have to take estrogen, but there can be certain safe ways. There are many herbal preparations. There are many herbal, everybody knows the flaxseed and sunflower oil, sunflower seeds. So these things are very helpful in, providing body certain amount of estrogens, so that the level of that desensitization of body should not happen. So I think, but one has to be very careful with the estrogen, the chemical estrogen. If you take the phytoestrogens, which are healthy type, which are known over our Ayurveda, then it is all right. But one has to take, you can't take estrogen for very long time, and you cannot, estrogen is not safe. So one has to find certain safe ways to, but certain women have very harsh, menopausal, so they have to take short-term HRT, and then it is required. Otherwise, quality of life can go down.
Ma'am, can you make me host and stop sharing the screen?
Yes. If there's no more question, then
1 second. Just you make me host because sir wanted to talk.
How do I make you host? Once I stop sharing, I think you are the host.
No, you